All right, if you've got a biology exam coming up, you are in exactly the right place. Don't sweat it. We're about to do a super fast, high yield rundown of the mitochondrion, covering all the essentials you need to know to crush those questions. Let's jump right in. So, we've all heard this phrase, right? The powerhouse of the cell. But what does that actually mean? What's going on inside this tiny organelle that gives it such an epic title? Well, that's the big question we're here to answer. Our tour starts right at the edge. Think of the mitochondrion like a high-security building. It's got two borders, two membranes, and you absolutely have to know the difference between them. Trust me, examiners love to ask about this stuff. Okay, first key fact to lock in your brain. The mitochondrion has a double membrane, two layers. This structure is totally fundamental to how it works. But here's the kicker. These two layers are very, very different from each other. And here is that critical difference. On one hand, you've got the outer membrane. Think of it like a chain-link fence. It's fully permeable because of these protein channels called porins, which let all the small stuff just waltz right through. But the inner membrane? Whoa, that's the VIP section. It's selectively permeable, like a bouncer at a club. It decides exactly who gets in to where the real action, the energy production, happens. So we've made it past the outer fence and through inner security. Now we're inside, where we get to see the mitochondrion's really unique internal landscape. You'll immediately notice that the inner membrane isn't smooth at all. It's highly folded into these complex, wavy structures called cristae. And these folds aren't just for looks. They have a super important job: to massively increase the available surface area. More surface area means way more room for the machinery of energy production. And all that extra surface area is crucial because it is literally packed with the tiny little factories that generate the power. This is it. This is where the magic happens. The very reason we call this thing the powerhouse. Studied all over the surface of those cristae are these tiny, well, they look like lollipops. It's a fantastic visual to remember for your exam. These little energy lollipops lining all of the inner folds. Now, while lollipop is a great way to picture them, the official scientific name you'll want to use in your answers is oxosomes. That's the term that's going to get you the points. Okay, lean in for a second because this is a huge pro tip for your exam. Test makers love to use synonyms to try and trip you up. So while you know them as oxysomes, be ready to see them called F1, F0 particles, elementary particles, or even Fernand-Morin particles. Know all four names. They all refer to the exact same thing. So what do these oxysomes actually do? Well, they act like microscopic turbines. As particles flow through, they spin, and this spinning motion powers the creation of ATP, the main energy currency for the entire cell. This little machine is the power in the powerhouse. So we've seen the membranes and we've seen the energy factories. Now we're journeying into the very core, the innermost part called the matrix. And, it's hiding a pretty surprising secret about how independent this organelle really is. Get this, inside the matrix, we find that the mitochondrion has its own circular DNA, just like a bacterium. It has its own RNA, and it has its own 70S ribosomes, which are actually different from the 80S ribosomes found in the rest of our cells. It's a complete, self-contained genetic toolkit. And because it has its own DNA and its own little protein-making factories, the mitochondrion can do something incredible. It can replicate all by itself. It divides through a process called binary fission, whenever it needs to, without waiting for the whole cell. Whew, okay, that was a lightning-fast tour, I know. But we've hit all the key structures and functions. Now, let's see what you remember. It's time for a quick knowledge check to really lock in that learning before your test. All right, first question. Think back to that outer border. What specific feature lets pretty much anything small pass right on through? The answer is porins. Yep, these are the protein channels that basically turn that outer membrane into a sieve, making it fully permeable. Question number 2. What do we call those little energy factories we saw studded all over the crista? That's right, they're called oxysomes, and hey, bonus points if you remembered any of the other names like F1, F0 particles. Okay. Question 3, this is a detail from the matrix. What specific type of ribosome does the mitochondrion have? The answer is 70S ribosomes. And that's such a key detail because our own cells have 80S ribosomes. That difference points to the mitochondrion's very unique history. Question 4, what's the vocabulary term for those folds of the inner membrane, the ones that ramp up its surface area? Yep, the answer is cristae. More folds, more cristae means more surface area for those oxysomes to make ATP. Simple as that. And for our final question. Thanks to having its own DNA and ribosomes, how does a mitochondrion make copies of itself?
The answer is binary fission. It literally just splits in two, just like a bacterium would. This autonomy is one of its most fascinating features, for sure.
So, we'll end on this thought. If the mitochondrion has its own bacterial-like DNA, its own bacterial-like ribosomes, and it divides by itself, what does that suggest to you about where it might have come from billions of years ago? It's a fascinating question to think about. All right, good luck with your exams, and thanks for tuning in.